Hello, and welcome to our session titled The Value of Genetic Research on Childhood Psychiatric Symptoms for Adult Outcomes. My name is Joanna Martin, and I will briefly introduce the session topic. We will then have 4 talks from a series of excellent speakers and conclude with a discussion led by Christel and Jordan. We know from decades of epidemiological research that there is a complex interplay of genetic and environmental risk factors that lead to psychiatric symptoms across the lifespan. It is also well known that there are worryingly high rates of mental health problems in children, and that these frequently persist into adulthood and can also be linked to secondary mental health problems like depression and substance misuse later on in life. In fact, a large proportion of adult psychiatric disorders onset early on in life, in adolescence and young adulthood. Therefore, studying childhood psychiatric problems will necessarily have important implications for understanding psychiatric disorders in adults. It is now well established that there are vast shared genetic risks across childhood and adulthood onset conditions. However, genetic studies of childhood psychiatric disorders are lagging behind in sample sizes and discovery of risk loci. This plot shows the largest available GWAS for predominantly child-based attention deficit hyperactivity disorder and autism spectrum disorder compared to adult-based bipolar disorder, major depressive disorder, and schizophrenia samples. Emerging evidence also suggests that early onset and persistent psychiatric disorders may be associated with a higher genetic loading. So there's a great need for more genetic studies of psychiatric problems in childhood and those with an early onset, for example, of anxiety, depression, and eating disorders in adolescents. We will next hear from 4 excellent speakers who will discuss these issues in greater detail. Our speakers are Henrik Larsson, Ditte Demontis, Kwong, and Sarah Medland. Afterwards, please join us for our discussion if the time zone suits you.
Hi, I'm Henrik Larsson. Thank you very much for inviting me to present on how childhood psychopathology links up with psychopathology in adulthood. So key findings from several epidemiological studies of childhood psychopathology have demonstrated that comorbidity is the rule rather than the exception, and also that the developmental course of psychopathology from childhood to adulthood involves both continuity and change. Because of that, an important goal of psychiatric genetic epidemiology is to identify the genetic and environmental underpinnings of both comorbidity and developmental stability and change. Many of us have used both quantitative and molecular genetic studies to produce estimates of bivariate genetic correlations across different types of psychopathology. The available genetic correlations for major psychiatric disorders is to some extent summarized in a recent review by Smoller et al. In molecular psychiatry. So what you see here in this table is genetic correlations from pedigree or twin studies below the diagonal, and genome-wide SNP-based genetic correlations above the diagonal. And one major explanation to the lack of numbers from twin studies that you can see below the diagonal is that most of these studies have used symptom or trait-based measures of psychopathology rather than diagnosis-based data. I also want to highlight that due to the high level of research activity in this field, this table might perhaps best be viewed as a snapshot of a living document that constantly is updated with new findings. The genetic correlations from genome-wide SNP-based analysis could, for instance, be updated with estimates from this recent publication in Cell from the PGC cross-disorder group. And several of the empty cells from the table I just show you below the diagonal could be filled with genetic correlations from this recent pedigree-based study. What you see here in the top left corner are relevant genetic correlations for several neuropsychiatric disorders. I also want to highlight some recent research from my own group. So we have conducted several register-based studies, familial congregation studies based on ADHD. And these could be used to fill in missing pieces in the column dealing with ADHD in the table before. And as you can see here, statistically significant familial associations are all over the place, but the effect size is particularly strong for early onset neurodevelopmental disorders such as ASD and intellectual disability. So what I've presented this far supports a quite broad hypothesis that most, if not all, forms of psychopathology across the lifespan correlate in part due to genetic factors. I would say that the key characteristic of more recent quantitative and molecular genetic studies is that they have moved from bivariate to multivariate approaches in order to advance the understanding of the genetic structure underlying psychopathology. The recent cross-disorder publication that I just talked about represent one important example of how genomic multivariate approaches can be used to identify a more detailed description of the genetic structure underlying psychopathology, with findings supporting 3 more specific disorder clusters characterized by strong genetic sharing. We and others have during the past years used multivariate structural equation modeling of twin and sibling data to identify a general factor of psychopathology. So in this study, on this slide, we found that about 30% of the variance in parent ratings of ASD, ADHD, tics, and learning difficulties could be explained by a single general genetic factor. In this study, we found support for a genetically influenced general psychopathology factor that influence across major child and adolescent psychiatric disorders. And here's another example of how we in a recent study used multivariate structural equation modeling of twin and sibling data, but now using the general factor of psychopathology as a covariate. So here we basically conducted multivariate genetic analysis of ADHD and co-occurring psychiatric disorders before and after accounting for the general P factor. So what you see here are substantial genetic correlations between ADHD and neurodevelopmental disorders, internalizing and externalizing disorders. What you can see here are results after accounting for the general factor of psychopathology. So genetic correlations for internalizing and externalizing disorders attenuated substantially, whereas the genetic correlation with neurodevelopmental disorders remained similar. Based on what I have presented this far, it seems like a genetically influenced general psychopathology factor is associated with many psychiatric conditions across the lifespan, in addition to several specific disease clusters characterized by strong genetic sharing. So what are the efforts needed to resolve some of the remaining knowledge gaps? Well, I'm tempted to argue that at least one important way forward is to conduct more longitudinal studies with data from childhood to adulthood. We all know that a substantial number of twin studies already have explored stability and change of psychopathology in childhood and adolescence, but more large-scale twin studies with adult follow-ups are needed. Studies that associate genetic instruments for adult psychiatric conditions with early indicators of childhood psychopathology represent another useful approach to move forward. There are still few longitudinal studies of the general factor of psychopathology. We have demonstrated that parent-rated general psychopathology in childhood predicts self-reported adverse outcomes in adolescence, primarily due to genetic factors. And a recent paper in JCPP have demonstrated that the stability of the general P factor is largely genetic in origin and correlates with general genetic factors in adulthood. As far as I know, there are still only a few genetically informed trajectory-based studies, but with important examples to learn from, such as a recent twin study on ADHD in JAMA Psychiatry, and research findings that will be presented in Alex Kwong's talk in this session. I'm finally arguing that there are too few large-scale genome-wide consortia with longitudinal data. But again, we have important examples to learn from, such as findings that will be presented in Ditte Demontis' presentation in this session on ADHD, and also past and ongoing work conducted in the context of the EAGLE consortia. So in conclusion, childhood psychopathology links up with psychopathology in adulthood via general and specific processes underpinned by a complex pattern of genetic factors. And examples of future directions in order to move the field forward involves large quantitative genetic and genotype samples with detailed longitudinal information on psychopathology, ideally both symptom and diagnosis data, combined with innovative multivariate and longitudinal statistical approaches. Thank you very much for listening.
I would like to talk about differences in the genetic architecture of childhood persistent and late diagnosed ADHD. So large sample sizes increase power to detect variants associated with the general liability to ADHD, and we have seen that in our latest GWAS meta-analysis. However, genetic heterogeneity might exist among ADHD cases. And one thing that might create this is that we know that approximately 2/3 of children diagnosed with ADHD will continue to experience ADHD symptoms during adulthood, referred to as persistent ADHD. And we also know that around 1/3 of individuals will grow out of the symptoms. And also some individuals are not diagnosed with ADHD before adulthood. So to evaluate the extent, so the aim of this study is to evaluate the extent to which the genetic architectures differ between childhood persistent and late diagnosed ADHD. However, it's very important to mention here that per definition, ADHD is a childhood onset disorder. So it is assumed that even though individuals are diagnosed in adulthood with ADHD, we assume that they actually experience symptoms during childhood. So for this study, we used cases and controls from the eyesight cohort. So we had the genotypes and we merged them with a register file with information about when individuals have received a diagnosis of ADHD. Then we defined 3 groups. Childhood ADHD was defined as individuals being younger than 18 years of age when they received and have received a diagnosis of ADHD. Then persistent ADHD, we defined that as individuals who had received a diagnosis of ADHD when they were younger than 18 years of age, and also when they were older than 18 years of age. And then finally, the last group, late diagnosed ADHD, and we defined that as individuals who got a diagnosis of ADHD when they were older than 18 years of age. So in the childhood group, they are younger than 18 years of age. So it consists of individuals, or some individuals that will continue to have ADHD in adulthood, and also some that will grow out of the symptoms. So here you can see the distribution of males and females. So as expected in the childhood group, we saw a clear overrepresentation of males compared to females. And this male-female ratio was also significantly higher in this group when compared to the other 2 groups. And we saw, especially for late diagnosed ADHD, that the sex differences is much more equal. And this is also in line with what we know from the literature. And here you can see the results from the GWASs of the three groups. So we found genome-wide significant loci for late diagnosed and childhood ADHD, but not for persistent. And this is also expected. We don't have power. We had only 1,400 persistent ADHD cases. But maybe something quite interesting is that the locus on chromosome 7, the FOXP2 locus, which we have discussed a lot in relation to ADHD, actually in ISAC seems to be driven by the late diagnosed group. Then here are some SNP heritability estimates. We found the highest SNP heritability for persistent ADHD, then late diagnosed, and then childhood ADHD. However, I'm not completely sure about these prevalences, so some cautions should be taken with respect to these estimates. And then we estimated genetic correlations using the genotypes and GCTA among the three groups. And as you can see here, we only found a moderate genetic correlation of 0.64 between childhood and late diagnosed ADHD. And it could be argued here that some of this might be caused by the childhood group also including individuals that will grow out of the symptoms. However, this argument doesn't hold when we look at the genetic correlation between childhood and persistent ADHD, which is 0.82, so quite high. And we also did find a quite high genetic correlation between persistent ADHD and late diagnosed ADHD. And we wanted to look further into these potential heterogeneities. So we looked at the ADHD polygenic risk load in the groups. And here you can see the normalized polygenic score of the entire sample. So we have normalized the score so that the mean is 0 and the standard deviation is 1. And then the mean of a subgroup in the sample tells how many standard deviations the average of that subgroup is away from the center. And you can see here that we did find a higher polygenic risk load in the persistent group compared to childhood and the late diagnosed group. And you can also see it on this figure. You can see the beta values and from the multiple regression. And you can also see that we found a significant higher polygenic risk load for ADHD in the persistent group compared to the other two groups. And it also makes sense that the group of individuals or that individuals that will continue to have symptoms in adulthood also have a higher enrichment of ADHD risk variance, at least when compared to the childhood group. And here you can see the odds ratio within each decile, and you can also see that it is highest for the persistent group. And then we also look at genetic correlations of the ADHD subgroups with ADHD symptoms. And for ADHD symptoms, we use the GWAS meta-analysis of ADHD symptoms in the general population based on more than 37,000 individuals. And those symptoms are hyperactivity and inattention. And you can see here that we did find a considerably lower genetic correlation of late diagnosed ADHD with both hyperactivity and inattention than we see for persistent and childhood ADHD. And this is also significant when we compare the genetic correlation of late diagnosed with the genetic correlation of childhood ADHD. So what this tells us is that actually maybe a part of the reason why some individuals get a diagnosis late in life with ADHD, it might actually be that those individuals are less genetically predisposed to be hyperactive and inattentive compared to individuals with childhood ADHD. Then we also looked at comorbidities in the registers of other psychiatric disorders, autism, schizophrenia, bipolar disorder, and depression. And you can see for the childhood group, the highest comorbid condition is autism. And for the late diagnosed group and also for persistent ADHD, the highest or the comorbidity that is most prevalent is depression. However, it could be argued here that Of course, we don't see a high comorbidity with depression in the childhood group because they're simply just not old enough in order to get a diagnosis of depression. But when we look at the genetic correlations, you can see them here. We looked at genetic correlations for various psychiatric disorders. We can see also at a genetic level, we do see a higher genetic correlation for both persistent and late diagnosed ADHD with depression than the genetic correlation of childhood ADHD with depression. And we do see the opposite pattern when we look at the genetic correlation with autism, where we see a significantly higher genetic correlation of childhood ADHD with autism than we see for late diagnosed ADHD with autism. And we also wanted to look at this also by doing polygenic risk score analysis. And here you can just see that we saw the same pattern when we did polygenic risk score analysis for depression, where we see this enrichment in the polygenic risk score for depression in late diagnosed and the persistent group compared to the childhood group. However, we're in the process of generating new polygenic risk scores for autism. So unfortunately, I don't have these results to show you yet. And then finally, we did a rare variant analysis. We have a whole exome sequencing data for a subset of the individuals, and we defined an allele as being rare if it had an allele count less than 5 in the combined sample of ISAC and NOMAD. And we tested for the load of protein truncating variants and synonymous variants in different gene sets. So a set of evolutionary highly constrained genes with high expression in the brain, and then a set of genes enriched with de novo mutations in individuals with neurodevelopmental disorders, and then an intersection of de novo and highly constrained genes. And then we have these gene sets for comparison, which are not, these gene sets are not high-risk gene sets. And this is a set of low evolutionary constrained genes and highly constrained genes, but with low expression in the brain. And here you can see the results from multiple regression. So you can see the increased load in the childhood late and persistent groups compared to the controls, and also pairwise comparisons between the groups. And what we found was, you can see here, consistently a lower polygenic load of protein truncating variants in the late diagnosed group compared to the childhood and the persistent group. However, these gene sets are of course highly overlapping, so it's not independent results that we do see here. And when looking at the synonymous variants, and also when looking at these protein truncating variants in the two gene sets that are not high-risk gene sets, we do not see the same pattern. But when looking at the p-values, it was only in the childhood group where we do see a significant enrichment of protein truncating variants in these high-risk gene sets in childhood ADHD. And we see a borderline significance for protein truncating variants in highly constrained genes in the persistent group. And when looking at these pairwise comparisons, we do not find anything significant. However, if we might have had more power, it could have been that we actually would be able to see that we do see an increased load of protein truncating variants in the childhood ADHD group compared to the late diagnosed group. And then finally, the acknowledgements. I would like to thank all the people from ISAC, and especially Vera, who did a great job analyzing these data, and also people from the Broad and Tatiana for sharing her summary stats from the GWAS of ADHD symptoms. So thank you very much for your attention.
Hi, good morning, good afternoon, or good evening, wherever you are in the world. My name is Alex Kwong from the University of Edinburgh and the University of Bristol, and I'm just going to be talking to you about how we can use genetics to examine longitudinal developmental traits over time. Really, this is just an extension of some of the talks that we've had this morning, or this afternoon, this evening. And what I'll do is I'll just be giving an overview of how we can use genetic contributions in the form of polygenic risk scores to really understand more about the genetic architecture underlying developmental traits. So when we talk about traits changing over time, this could be anything from physical conditions such as height and weight, to cognition, so educational attainment or IQ, or psychological or psychiatric conditions such as depression or anxiety. When we're talking about how traits change over time, we're interested in how people change across certain periods or transitions of their lives. So, for instance, childhood to adolescence, adolescence to adulthood, and beyond. But it's quite important to establish that these changes are not always stable, and you may get certain periods of growth which are more unstable, where you may have greater rises in a particular trait, such as early childhood, where you see a large increase in height and weight, for instance, and then for psychiatric conditions, you may see an increase in depression or anxiety in the early adolescence or early adulthood stages. And what we can do is essentially estimate traits and how they change over time using trajectories. This is typically done in the form of multi-level growth curve models, latent class or growth mixture models, or through forms of structural equation modeling. And what we can see is from the existing literature in developmental psychopathology is that these changes in these traits or changes in trajectories are partially heritable alongside a host of environmental factors. There's a great paper by Laurie Hannigan in 2017 in Psychopathology Review, which outlines a meta-analysis of this through twin research. And I think what the key point that I want to outline this talk is that through advances in genomic research, mainly through the PGC and through other bodies or consortia, we've been able to develop polygenic risk scores, which can be incorporated into these models, into population studies or longitudinal studies, so we can examine how polygenic risk scores can affect trajectories and start to ask really interesting questions. How are they associated with the onset of a particular condition, or how do they maintain a particular condition as well? So, as a quick primer for people who may be on this call or watching this who are not familiar with the developmental trajectories literature, what essentially we're doing is we're taking repeated assessments from an individual over time and sort of plotting a trajectory or a line of best fit to understand how they change over time and via the responses they've given. So here we have three different people, the red person, the green person, and the blue person. And what we can see is that the red person gave relatively high responses across different periods of development, so across childhood, adolescence, and adulthood. In this example, the green person gave sort of lowering responses of depressive symptoms, whilst the blue person gave relatively low throughout all the periods of assessment here. And what we can do is take these individual level responses and put them into more population group approaches. So one thing that's commonly done in developmental psychopathology is to estimate latent classes or growth mixture models based upon these individual level responses. And here what we do is we essentially just put people based upon their responses or groups of people who are similar into specific groups. So for instance, if you had a certain number of people who all responded similar to person 2, they may form that very high trajectory on the right-hand side in Model A under group-based models, where they were sort of a starting high and consistently high trajectory. Whereas if we had 50 or 100 people who were very similar to person 3 in the green line, they may form that sort of starting high but then getting better over time. And that's one approach we can use. The other approach is to do sort of hierarchical linear modeling or multi-level growth curve modeling, where we just estimate a population level trajectory and people are allowed to deviate from that trajectory. So on average, everyone will look like this, but underneath that population level trajectory, people will have their own trajectory, which might be completely flat, or it might be quite zigzag, or it might start very high and then go down. But on average, across the population, so maybe boys and girls, or individuals of high depression polygenic risk scores or low depression polygenic risk scores, they'll take separate trajectories, and we can see that for, on average, each population. So what I'll do now is just give you a very brief summary of some of the research which is coming out that's using polygenic risk scores to examine developmental traits. And what I'll do is I'll start here with some work led by Alexander Lussier from Harvard Massachusetts General Hospital, who used the Avon longitudinal study of parents and children, a birth cohort in the southwest of the UK, to examine developmental trajectories of depressive symptoms from the ages of 4 to 16, so very early childhood to mid to late adolescence. And they found, so Alex and his colleagues found that using the latest NDD polygenic risk score derived from David Howard's GWAS, you can differentiate between minimal symptoms trajectories, so trajectories that are quite low in depressive symptoms, with those that are sort of onset in towards late childhood, and those that are consistently high across childhood and adolescence. And what this shows is that using adult GWASs to make polygenic risk scores for children can, show associations between minimal trajectories, those who are displaying minimal symptoms, compared to those who start showing symptoms across childhood. And I think it's very important to mention here, this does not imply that higher genetic loading for depression in the form of a polygenic risk score is deterministic or is in any way causal. It's more reflecting a very complex biology or a complex underlying mechanism that interacts or is mediated by the environment, which we don't know fully enough about yet. But it's basically just showing that we can use polygenic risk scores from adult samples to help understand more about developmental psychopathology, especially from transitioning from early childhood to adolescence. Some similar work coming out of Cardiff University, led by Professor Frances Rice, has also found quite similar results when using the Ausbach study, but over a different period of time. So Frances and her team were able to estimate 3 trajectories of depression between the ages of 10 and 18, so sort of middle to later childhood to later adolescence. And in a sample of 7,543 individuals, they found that the MDD, the schizophrenia, and the ADHD polygenic risk scores were associated with an early adolescent onset trajectory, so that blue line there. But they found that only the MDD polygenic risk score was associated with a later adolescent onset trajectory. This is quite important because it implies that different genetic underpinnings for different traits may have differential impacts on when they start to onset and when they start to exert their influence. So again, not saying that these polygenic risk scores for schizophrenia, ADHD, or MDD are causal, but are in fact reflecting a complex interplay between the environment and genetics, which with these kind of studies can help underlie or examine some of the underlying biology. And I think what this is quite key in showing is that there is some good evidence here showing that major depressive polygenic risk scores take a very complex pathway. So it's not just that if you have the predisposition for higher genetic loading for a depression polygenic risk score, you take one trajectory. Actually, you can take a multiple number of paths. And actually, that's quite important going forward because it just doesn't make it as simple to say that having higher genetic loading for depression is going to put you on this path. It's actually going to reflect a much more complicated system, which is going to require further research. And this brings us on to the final thing we're going to talk about, which is how you can use the MDD polygenic risk score to examine changes over time, but also go beyond traditional cross-sectional research. So we found that the MDD polygenic risk score from David Howard's GWAS was associated with self-reported depressive symptoms at each of the 9 occasions in which we assessed depressive symptoms, and that there were stronger genetic influences as time went on and towards older ages, which might be expected. But actually, can you go beyond this cross-sectional analysis to utilize the repeated measures and the correlation between the traits or between the measures to sort of improve the signal, to improve the associations? And what we found is that you can go beyond just simple cross-sectional research to actually estimate trajectories which can inform the severity and the rate of change of depressive symptoms that are associated with the MDD polygenic risk score. What this does is goes over and beyond the traditional cross-section research by quantifying how much of the polygenic risk score is associated with specific changes at specific times. And so what you can say is that the polygenic risk score may be kicking in or starting to influence development at a greater rate at the ages between 14 and 16, and that this is maintained throughout adolescence and early adulthood, whereas in traditional cross-section research, it's not possible to estimate within the same sort of model when things may be kicking in or anything like that. So actually what we've done is gone beyond the traditional cross-sectional research and to use repeated measures and trajectories to sort of improve the power and the precision of these association studies. So that's just a very quick whistle-stop tour of some of the research that's been going on looking at polygenic risk scores and developmental traits. If you are interested in these studies and you think you want to know more, you want to look and see what else has been done, here are some other studies that might be of interest to you, looking at different developmental stages and different kind of risk scores. The first is one by Benjamin Dominique in looking at the health and retirement study and looking at some G by E. Second is something where we looked at the depressive symptoms polygenic risk score and other environmental risk factors to see which are the kind of major predictors of how people transition across different stages of adolescence and early adulthood. And then some work led by Lucy Riglin in Cardiff has shown that the schizophrenia polygenic risk score sort of is associated with early onset mental health trajectories that also might be related to later mental health trajectories as well. So those are some things that might be worth having a look at if you are interested in this topic. So just to summarize, trajectories are able to help us examine the longitudinal nature of traits through the identification of key developmental phases and transitions through life, particularly through early life, through middle life, and later on. And through the use of polygenic risk scores derived from PGC and other consortia, we can examine and include these genetic markers into our developmental trajectory models to examine how they may impact on how things change over time. It's not just restricted to depression, even though that's what I've talked about here, and it could be for any other particular trait of interest. The overarching results from the majority of the studies I presented have shown that higher polygenic risk scores, so greater genetic loading for a particular trait, are frequently associated with higher or more severe trajectories. This implies that there's a strong genetic contribution to the onset and maintenance of these different traits, and this can also help explain some of the biology underlying what's going on from how people transition from low symptoms to higher symptoms, and if that continues throughout development. But it's very, very clear, and really cannot stress this enough, this does not imply genetic determinism or causality, or that having a higher polygenic risk score for a particular disorder means you're going to be determined to be on that higher trajectory or that lower trajectory or whatever. That's not what we're saying. Instead, it's more likely to reflect a really complex interplay with the environment as well, which we just don't know enough about yet, but we are looking to see in future research and look to unpick apart. Just to say that, a bit of a primer or a bit of a teaser to say that the next MDT-GWAS paper, we are looking at developmental trajectories as part of the downstream analysis, so keep your eyes posted on that. Likewise, you'll probably see many other studies coming out which are looking at different kind of polygenic risk scores and developmental traits, because I think it is quite an exciting avenue of work. If you are interested in looking at polygenic risk scores and trajectories, or if you have any questions about this talk which I've given, please feel free to ping me an email or get in touch with any of the other people talking in the session. Great, and thanks a lot.
Hello, my name is Sarah Medland. I've been asked to speak today about recruitment and methods to find participants. So obviously there are strategies we're all very familiar with. The three methods I'll be talking about today, social media, medication, and media appeals, are used less commonly, and I'll be illustrating each of these with studies that I've been conducting. So starting with social media, I used this strategy to run a study looking at nausea and vomiting in pregnancy. And the reason why we wanted to study this is because there's actually a strong correlation, phenotypic correlation, between NVP and depression and anxiety. So NVP is a risk factor for depression and anxiety. Depression and anxiety is a risk factor for depression. And children born of pregnancies with severe NVP have a higher risk of depression themselves. So in the literature, historically, there's some pretty interesting psychoanalytical interpretations of NVP. But our hypothesis was that what we were seeing there was actually genetic pleiotropy. So to test this, we undertook a social media recruitment where we recruited phenotyped and genotyped case control cohort. And we did this by approaching some Facebook pages that had a large number of followers and asking if they would be willing to post a link to the study or a short blurb about the study. The study questionnaire was completed by 1,700 women, and we were able to really recruit quite a severe case control cohort there. We then also recontacted cohorts with existing genotypes and collected phenotypes, and collaborated with groups in Europe and the US. We ran a GWAS meta-analysis and then LD score, and we were able to show that MVP was indeed genetically correlated with depression. So this has implications for treatment in terms of monitoring and risk of depression during pregnancy. And there's a couple of real advantages to the method here. So we had no budget for this study when we started. So we were able to do that first initial recruitment with a budget of around $100. This enabled us to convince others to come in and collect the same phenotypes or to join the meta-analysis. And it led to a grant to go back to these participants and collect DNA and genotypes. So some things that we learned that you need to keep in mind is you really can't control what support groups or influencers will post, and you do need to have a conversation with your IRB about this before you start. The other thing is tagging and commenting is really a problem. So here we have an example from our national media service where they're actually doing a recruitment to obtain lived experiences from people with disabilities. And we can see here a selection of comments where people are tagging their friends, including their full names, and making suggestions that these people would be good for the study, which actually suggests that these people do have a disability, although they themselves have not consented to be publicly named. Okay, so the next strategy I'll talk about is a medication-based recruitment. And in Australia, we can do this because we have universal healthcare and prescriptions are subsidised. So prescriptions that are subsidised are recorded in a database, and there is a section of that government department which runs a mail-out service. So after an extensive approval process, it's possible to send study information to individuals who meet criteria, although it's important to note that researchers do not receive information about who gets the letters. So we use this method to recruit a cohort of Australian children living with ADHD, and we thought this would be possible because there's really quite strong monitoring of ADHD medications in Australia. So we had inclusion criteria around age and also the number of prescriptions that these children needed to receive. And the study information was sent to the parents or caregivers of the children, and the letter contained a webpage link that they could go to find out more about the study and to participate. So the study ended up, the questionnaire ended up being completed by 2,100 caregivers, and the proportion of males and females and also demographics and subtypes was representative of the population. So consistent with previous studies, we had a high degree of multiplex families and a strong history of ADHD amongst the parents of these children. This study method was really quite efficient. So the total budget here was $30,000. Once again, we were able to leverage this to obtain a grant to go back and genotype and these children. So things to watch out for: the approval process really took a lot longer than we expected, and there was a high volume of calls and emails about the study. I personally returned every single phone call, which took quite some time. The other thing to know is that with this type of study, the responses come in thick and fast, and if you have any type of IT outage or problems during that first week, you're really going to lose the recruitment. Okay, lastly, a media-based appeal, and I use this in a study focusing on bipolar disorder. So the aim here was to recruit, once again, a cohort of individuals living with bipolar. So we worked with a group of core participants who were willing to share their experiences in the media, and we had a physical media launch followed by study invitation letters. So the media launch was covered quite well, both in print, radio and TV media, and there was some sort of unexpected wins. So we had a TV station that made quite a nice infographic, which was actually shared and viewed over 35,000 times. So the online questionnaire to date has been completed by 5,500 participants. Almost half of them completed it on a mobile device. And we see, as you'd expect, the kind of experiences that participants are having. So this study was able to reach a nationwide sample, and we were able to reach participants that traditional clinic-based recruitment wouldn't reach. So quite a large number of people from smaller towns and regional areas. So the strengths really are that it's a large nationwide representative sample, and things to remember are you really can't control the media. So on the day of the launch, there was actually a terror-related incident in one of the most populated states, and that led to very little media coverage of the study in that state. There's also quite a high volume of phone calls and emails, so it's important to be prepared for that. Just to wrap up, I'd like to make a couple of points about online survey-based phenotyping. The first one is that it's really worth considering separating the consent and contact information from the phenotyping. We actually use 2 different softwares for this, and we just port the study ID between. It's also really important to think about including some ID checking questions in your survey. So it's very common that participants will forward the emails with their unique links to their friends to enable their friends to participate. So by having these ID checking questions, you can check that the person consenting is the person giving phenotyping information. Lastly, when thinking about collecting DNA by mail, there are quite a number of vendors that do provide systems for collecting saliva. It's important to think about the physical size of the tube compared to the physical size of mail in the country that you're working within. Think about the design. And then the last point I'd really like to suggest is that you have a conversation with your vendor and ask to get the tube supplied without the clamshell packaging. Remember that that packaging is not recyclable, and when you're doing a large study, you can create quite a large number of items going direct to landfill. So we want to minimize that wherever possible. Okay, thanks very much. And if you have any questions, please feel free to email me.